So, welcome everyone. I'm Sharon Terry, President and CEO of Genetic Alliance. Delighted to be with you all today. We're going to have a very interesting and special invitation here for rare disease advocacy organizations to co-design a new platform for identifying drug targets, beginning with human validation. And we have the pleasure of hearing from and working with Stephen Friend. Stephen is a longtime friend and a former board member of Genetic Alliance. He's both a clinician and a scientist, and he's been focusing on genetic resilience, which he'll tell you about. He's currently the president and co-founder of the nonprofit For You and Me, and he's a visiting professor of connected medicine in the Department of Psychiatry at Oxford University. I met Stephen when he was in his co-founding of Sage Bio Networks, and we worked together on a whole host of things. And so I'm delighted to be back working with Stephen and to give him the floor today. So, Stephen, over to you.
Thank you very much. An extra special thank you for the intro, but also for the links we've had. I'm going to go ahead and share my screen. And I have about 15 minutes that I want to use to set the stage. But as Sharon said, the real goal is actually to have the dialogue afterwards. And so if you'll be kind enough to sort of buckle down and let me zip through these. The point is that we can get back to these slides in the discussion afterwards. So this is a long title, as usual. Exploring a new platform to find drug targets. That's pretty clear. Starting with human validation that couples the power of rare outliers. And that part, I hope, makes sense by the time I finish the talk, or at least we finish our discussion. I'm going to go from like, where does this come from? And the grounding on drugs that are discovered, even though I think some of you know it very deeply, to some aspects on genes and mutations. And sort of this is based on sort of like what we could learn from lower organisms. And so I need to zip that in before going, why do we think this might work, to what are we proposing, through to what Sharon talked about is at the end. It's sort of an invitation that will lead to a survey. So where did this start from? Well, I've been doing this stuff for a while, and over those years, decades, not years, the ability to work in genetics and dovetailing that with drug discovery and then more recently wearables and drug discovery has been sort of a constant interwoven theme. So it's something that has been sort of active in various people's minds. I highlighted these people who really helped shape how I think, and many of them work in lower organisms. But there was one thing that basically set this and a lot of what I do in place. When I was training as a pediatric resident, early oncology fellow at the Children's Hospital of Philadelphia, I had the chance to see a dad and a child, and in examining the child, that child had a retinoblastoma, but it didn't take great clinical skill to notice that the same left eye that was altered in the child, there was a removed eye on the left side of the dad. And this was one of the genetic forms of retinoblastoma. And it just seemed so hard to carry that sort of sense of what had happened. And that, with many other things, sort of propelled us to be thinking about how could you know more about that disease. This is like 81 to 85. And by 86, thanks to large groups of people at MIT and really many places, we had pulled together a group that was able to pull out the gene that was responsible at a time when not much of that was happening. Sharon, you'll appreciate the fact that Lou Kunkel, the same week in the same issue of Nature, had one on muscular dystrophy, one on retinoblastoma. And the reason I point that out is that here we are, 4 decades later, the children with retinoblastoma and Duchenne's, and basically all the children with thousands of other diseases, do not have access to, notice the important phrase I have, affordable therapies. So I'm not talking about, do you have a gene editing that your organization got lucky to pull together and now it costs hundreds of thousands? I'm really looking for how is it that we can get therapies that don't have that cost barrier. And this project is all about that. So to get there, this slide that could have 10 slides, I pulled it down to one because I wanted to keep this short, but also because I think you know all of it. But generally, these quote ideas that start these projects in drug discovery come from someone going, I noticed this or that, and it's in usually a narrow context. Sometimes it has nothing to do with clinical data. And all of this work zips through to the thing that I think you also know, which is by the time you get to phase two and looking at validation, is this really working or not? You've put an enormous amount of money, and way too often that comes up short in terms of actually, no, it doesn't work, or oh, it has this effect that you hadn't expected. And it's because of that. And one other thing that I'll bring back up a couple of times, the when you have a loss of function allele, which most diseases have, so where the mutation is knocking out the function, small molecules have an almost impossible job to actually bring that back to making that particular protein normal. So keep those two ideas in your head. And so what we've been working on for a while, this started with discussions with Phil Sharp, were what would it take to build a novel drug discovery platform where the targets are picked that are validated to have a beneficial effect upfront. The idea of those targets are done not by generating new genomic data, but existing. Could you make it so that the early work is guided, but not necessarily done by patient advocate groups? All the data in the compounds would be made open source, and the concept is to actually try to get the end cost, not something where an investor is trying to get a return on the money that they were kind enough to give, but they've always got to get some type of return, and try to make it so that there aren't patents that are sitting there requiring funds for that. And in terms of how to get there, this slide again is something that you're all too familiar with, this zip through to what I think similar to when we were finding a gene alter, there's sort of a sense of if we just had the roadmap, it would work well. And in that time, from back in the 80s through till now, that number of diseases known to be altered by specific genes has grown to this enormous number with that gap that I talked about. So what is Stephen talking about? The reality is that when you go to any lower organism, whether it's in yeast or in C. elegans, Drosophila, mice, and in fact it applies, we think, in humans, there, when you have a normal genome and you're looking at a trait like mobility, and there's an altered gene, sometimes those altered genes are a yes/no switch to a phenotype. And most of the time, if you have some other altered gene in the genome, it usually doesn't affect that no mobility, but occasionally, very rarely, when you have an altered gene A and you alter something else that is suppressing that, you can revert that. So this is called second site suppressors. Second site suppressors, as opposed to modifiers, where modifiers are changing the symptoms slightly. So there are lots of modifiers that we know, but if it's only changing it slightly, it's probably not going to make a massive difference to the person if you were to be able to do that modification. So we call these suppressors. They're called second site because of the fact that we're particularly looking for another protein target that isn't the first that we can break. So the concept is, out of all the reasons for having those abilities to shift the phenotype, there's a subset of those which are actually have another protein, and by breaking that other protein with a small molecule, you can sometimes have that basically fix the first. So this is second site suppressors. And what is known is that across all of these model organisms, a large fraction, and I mean a very large fraction of every protein with an associated phenotype actually has one of those second site suppressors if you can do what we can't do in humans, which is sort of a saturation mutagenesis. So what we want to know if those second site suppressors are so common throughout evolution, could we, could these second site suppressors represent good targets for small molecules? And how could we find them? How could we find who had those? Because they'll show up in people that are healthy. So you can't go and say, oh, this person has this disease and this. So you're sweeping through looking for people who apparently don't have anything wrong. I'll show you later, or in very rare families where many, many people have a known mutation with a known phenotype who have an odd outlier in those families. So those are our two processes. And the question is, why not find a way to identify those who have those? So how do you do that? Well, just one quick thing before there is if you might go, give me an example of a drug being able to come from that. Helen Hobbs, an amazing woman, force of nature in Texas, had studied some rare African American families and found some who had a genetic trait where even though they had high cholesterol levels, did not get heart disease. And out of that came an antibody, not a small molecule. You can build small molecules or antibodies. We can talk about that later. But this is a really nice example of doing some genetics, finding something that didn't match, and sort of was an impetus for us going into this area. The reason why we think that it is possible is that about 10 years ago, Eric Schott and I, who are working together now on this along with another, did a proof of concept effort to look at second mutations. And this article, and there's a TED Talk that's got over 1 million hits on it that you can go find called The Resilience Project. We swept through existing 500,000 or more genomes from basically everything at 23andMe, everything that was at BGI, et cetera. And we looked for changes that were sitting in a core set of 162 genes with their various alleles that we could, with talking to genetic counselors and others, say, here's one that seems to be fully penetrant. I've never found anyone who had that particular mutation and lived past the age of 10. So we did that so we didn't have to go back and find medical records. And out of that list and out of that effort, we found more than a dozen individuals who actually fit this bill of they were apparently healthy. We couldn't recontact them, something I'll go into later, but at least we saw that there were people, and we went and redid the sample. I mean, there are all these reasons that took what looked to be about 1,000 of these all the way down to just around a dozen to make sure that it wasn't just some artifact. So we're pretty confident those things existed. So for a while, we tried to get GlaxoSmithKline and Novartis and other companies to get in. And of course, what they're interested in is profit. That they don't want to go after something that has a small market. Thank you very much. That would be interesting. And so we couldn't get traction. So several things in the technology have changed recently. And so we decided to start this Resilience Project 2.0. And this time, asked, could we build a platform that was potentially able to help a number of rare genetic disease groups? Because the first part, and sweeping through large genomes or looking in outlier families, is fairly scalable. The difference between doing it for 20 and 200 is not actually that much different. And it starts by identifying resilient individuals, and then it assumes that you would like to have a set of coordinated teams. And our point is that I think if we did this and just found the second site suppressor, nothing would go on more. So our sense is the right thing to do, and this is what we've been doing for about a year, is to build a coalition. Sharon, you'll recognize, the only way we think we can get things done is to have everyone else help us do it. And so build out teams that are looking at finding those resilient individuals, other sets of teams who use AI and machine learning to go of all those loss of function alleles that sit in a given individual. You have to sift through somewhere around less than 200, but go, is this a candidate? Is that? So there are new tools I'll show you that we use for that. Then you've got to validate those. Then you have to go and find people who design probe compounds, do the clinically. So we've basically been going and talking to, you would expect, the normal large nonprofit foundation saying, is it something, would you be willing to put a syndicate together to fund this sort of large platform that many people could use? So that's what we've been doing. The 2 points on a little drilling down to, for those who know it more, to find those resilient candidates by screening. We're hoping to go through now 5 million genomes. So between all of us, between UK Biobank, between Qatar, we've been talking and actually been screening. So I think we can get to 5 million individuals, so 10 times more that we could sort of, but also like a project that we have in Jordan on retinoblastoma, sometimes, particularly in consanguineous populations, you can have families that are pretty extended and similar to the work we did with Wash U on Alzheimer's. You can sometimes do the finding of a rare individual in a family. And that is just as powerful. If you can find the right individual in a rare family, it's a second pair to sort of getting started. But then it's going through using functional data and cell lines to get what is in the center of this slide, which is knowledge graphs, which allow you to then stack order what are the most likely second site suppressors through to doing the assays to the validated targets. And here, and these are slides we can go back to, the goal is the more data you can bring in from existing. So if there are groups who have been studying a particular disease for a while, we can take that data and drop it into the core data lakehouse. And the more data that we have, the better it gets. So, for example, Eric Schott, working at Pathos, has millions of records on patients and outcomes in cancer and has single gene sequence, blah. So this helps us to bring in data in order to look at relationships. So knowledge graphs basically talk about, when I see this protein doing this, does it look like it is connected to that? That's one of our ways for doing the sorting that we couldn't do. We have 13 teams that we've been putting together, people like Jeff Ginsberg and Tom Maniotis, to people at the Broad, over to Bob Horowitz and MIT. And these are people who are acting as our core expert lead for these teams, saying, I will help guide what it is that you're trying to do on these. Nadia Rosenthal at Jackson Lab, Aled Edwards at the Structural, Corey Bargman, who used to be at CZI. So we've been going and finding people who really know what they're doing in each of these areas and going, can you help us? Could you bring this together and help us design what we need? And now to the, I think, 2 slides to go. So remember that slide I had before. What we're now doing, and Maddie is on the phone, Maddie Sorensen is helping me here for you and me, is we're going through and priming the pump. We're going through, going back to John Wilbanks and Megan Dorr and getting a consent that would work for finding someone who is healthy, and you're about to tell them that you'd like to study them more and they have something they didn't know. That's a very tricky consent, right? You have to think very hard about that. But you may know Megan and you may know John Wilbanks. These are serious experts on that. And so we're getting that ready while we're, and this is what I'll talk, the invitation is around looking at rare disease advocates as co-founders to sort of get the sense of how many groups might be willing to work like that. We've been completing these searches of various biobanks with a core set of genes that we had before, just to test how easy is it to do it now. And I mentioned the rare outliers in those families in Jordan. We've got them all, the DNA analyzed, and we're just trying to find a place we'll do the sequencing on that. The discussions, there are several groups who know how to do the assays using CRISPR induced vectors to look at combinations of what happens to phenotypes in, for example, in the Netherlands. And then, as I mentioned, Al Edwards has offered us some slots that if the candidates look strong enough. So the point is, we're trying to get these all done and not do one to the next to the next. And in line of that, this webinar is around the following. We think that it is worth getting a sense of the appetite of various rare disease groups to work in this way of being co-designers, co-founders. So we're looking for sort of the set that step up of founding rare disease groups, working on diseases, which have strong phenotypes and high penetrance. We can go into it later, but if it is something that doesn't have an obvious phenotype, and more importantly, it has variable penetrance to start with because of a bunch of modifiers, those we think will get in our way to doing the first proof of concepts. And so we're looking for rare disease groups, easy to be these co-designers with us to search for extreme outliers, who we will work with to identify we're able to do the work when we find the individual. Jeff Ginsburg has offered to potentially use the clinical trial center at the NIH to work up the family and other members because it's really helpful to do not just the one individual. So some role that the NIH may play in terms of the clinical center is, I think, being contemplated, but it could be that comes about. And so this webinar was meant to sort of have a dialogue. Sharon thought it was a good idea to sort of have a group discussion. And depending upon how that goes, we've got a flyer and a survey ready to send out. So I'm going to go off of that, go off of that. For now, I'll stop sharing, but I'm glad to go back to any slides. But I hope, Sharon, that gave enough meat and didn't use up so much time that we can't have dialogue.
Yeah, absolutely. Thanks, Stephen. Yeah, so when Stephen came to me with this idea, and I know that he's been going along cooking with this for some time, and aspects of it are aspects that both Stephen and I have been interested in, for example, the openness of this kind of work. I thought it'd be a good idea for him to give you this background and for us to kind of pressure test some of this so that then he can send this survey and flyer out. We'll use Genetic Alliance's listservs and Google groups and so forth. So maybe reaching around 2,000 advocacy groups, 10,000 individuals that are in leadership positions. And so love for you guys here, kind of our almost focus group, to ask questions. And I want to say right up front, Stephen loves hard questions. He has a mind that thrives on rich dialogue, so really feel free to ask anything. And the other thing is no question is stupid. Lots of these concepts are complicated, particularly for those of us who have no scientific background, and so any question is really fine. So I'm going to open the floor. Allie, you have a question in the chat you might want to start with, and then let's go on.
Sure. Hey, Stephen, I think I kind of know the answer to this based on your last slide, but we'll do it anyways. So my question is that I work with a lot of groups where the mutations are largely de novo. There isn't really a familial component. They're largely gain of function mutations that are rising spontaneously. I kind of assume that this is not the sort of mutations you want to be working with, but I'll just let you answer the question of how much harder is this to do for mutations that aren't familial?
That was like a spectacular, not a good, but you know how people go, that's a good question. No, that was a spectacular starting question because it really is a complicated one. So remember that, and the short answer is maybe, okay, instead of no, that really is out. And the reason I say that is the following. What we need to have is examples where there's enough knowledge about a mutation and its consequences that someone who's carrying that, and I'm now going to say de novo mutation, right, is has a extremely, high penetrance and a phenotype. If that is the case, then I think that finding someone not who had that de novo mutation and did not go on to have that phenotype would be approachable in not in the sort of looking at rare outliers in family, that's obviously off, but in sweeping through the core data banks. And I get to introduce Yolanda Van Neuven, who's in the University of Massachusetts. Yolanda, I just want to make sure you can check one of my colleagues that I work with and has done a lot of work on second site suppressors, brilliant young researcher. But I would have pinged Yolanda, my helpline, right? I'd go, hey, Yolanda, am I blowing it? Yolanda, you want to help us here?
Yeah, so we've tried to find suppressors for a lot of different genes. And I think Stephen also mentioned this briefly in his talk. We do find them for almost everything for which we try. And so I think it's possible. It's not it's not easy, but that's another thing. It depends a little bit on the type of mutation, and I'd be happy to go into details about that if people are interested. But especially if it's not a 100% loss of function allele, we can find suppressors for almost everything.
So I think that the tools that we have would eliminate, as you correctly guessed, Ali, the looking for rare outliers in families. But I think our major pillar is that sweep through for people who are apparently healthy. And what we found before was when you go and you look, there was a person who had ataxia telangiectasia in the UK on that first screen, and they were a 40-year-old with a mild amount of ataxia. That gets really exciting. That's the type of thing where you go, that person shouldn't have that level. Do you know what I mean? So I think that we'll get back to you, Ali, but the first answer is likely and shouldn't be it shouldn't be an exclusion criteria.
Great. Monique, I see you unmuted.
My question, I'm new to this second site suppressor concept, so sorry if this is naive. If you've got the mutant allele that turns off the function and then you find a suppressor that overcomes that, I presume neither is necessarily binary. Could be, the shutting off could be, but do we see sort of a titration almost where almost like with cystic fibrosis, the various, hundreds, thousands of mutations, there's some that just need a tweak, right? It's not like you don't have the function of the protein. You just need enough. Is there sort of evidence where it's neither side is binary, the suppression or overcoming, but you just need to get to 2% or 5% and you see not only a second site, but a combination, obviously, of other sites that overcome the deficit.
Yeah, that has several components. We'll try to address them, but I want to first say that you're new to the concept of the second site suppressor, but it's clear that from your question that some aspects of the equivalent of how to blunt the effect of a primary mutation and the CF thing, you're familiar enough that I can sort of build off of that. So the CF story is one where those are modifiers of the primary mutation protein, right? So what those are doing is going, I want to help you refold the right way, okay? And so those are not examples of finding another protein in the genome that is your sort of toggle switch, right? So that on those, the ability that some mutations you'll allow to bring back into normal conformation and normal function is among all of the mutations that they have in cystic fibrosis, some it works well on and doesn't, and that whole theme you were talking about. For the second site suppressors, what we're doing is finding a mechanism by which the cell is sort of released from a dependency on that first protein. And when you release it in various ways, then you can open up you can improve the phenotype of lots of the mutations in the primary gene, including total loss of function.
Okay.
And so always titration on both sides are there. I'm going to move that to the side, even though you're correct on it, because it's not a major factor of what we're thinking about. But the most important thing is think of this as not trying to modify the primary, not trying to change the transcription or the translation, right? There are all these other things. We're working on a subset of all the suppressors that are second site, and their second site where if we disrupt the function, like Yolanda said, where you knock out the function, it'll go in the right direction. So that's and our sense is that that's about 20% of the individuals who may be a resilient candidate. So it's not 90. But if you do enough genes and you look at enough people, the fact that it's only 1 in 5 resilient individuals is not a negative. It's just you don't go on. You can't go on. Do you see what I'm saying? So in drug discovery, you're trying to find the thing that's going to work and not worried, I'm not going to go here, I'm not going to go there.
Just to follow on, if I could, Stephen, just to make sure I'm grounding myself correct in this concept, could this second site be modified by environmental factors or something, not necessarily by nature, but the nature of the genotype itself, but that there's some non-genetic component that is a mitigator or amplifier?
Yeah, let me back. That is a clear yes. Okay, so there's one short answer, yes. Okay, now let me go a little bit.
Okay.
But you got it. And there's an entirely different, much harder project that we're trying to get funding is what about the resilient individuals for whom it is their context and their environment that they're in that might be able to be modified. There's a whole other one. If this one is super hard, that one's even harder, but it's beautiful because then you aren't trying to find a drug. Do you know what I mean? So I want to say that your concept of are there things that aren't genetic alterations that could act? And in lower organisms, context matters, right? And so they're like, it's not just genes. And so know that. But the ability to scan and find those, right, has sort of tools that we may eventually evolve. But the easier case to look for, at least at the beginning, is can we find proteins in the resilient individuals that we can try to knock out and show that. And but I really like the question, and there's a set of people, sadly not enough funders, who would love to go down the non-genetic resilient path. And so I'm really happy that you brought up. Sometime we'll have a funder listening who will go, well, maybe we're interested. So we'll get there.
Other questions? Megan?
Yeah, thank you. So you're focusing on highly penetrant mutations, right? How penetrant do they need to be, and could it work on one that is it loss of function that we're looking at? No, a gain
of function, yeah.
Gain of function mutation that isn't as penetrant. Could it also work there, and you just want to start with the more penetrant ones?
First of all, I have to say, Megan is not unknown to me. I've had the distinct pleasure on a CCI funded project of working with Megan, and I'm used to her depth. So when I saw her on, I thought, I'll be disappointed if I don't have a question. Also, I have to say, Megan has seen some of this when I was trying to figure out what we were going to do, and in getting here, Megan, thanks for thinking of that. So now to your two questions. One had to do with why do you focus so heavily on full penetrance, and the second one is the primary mutation a gain of function? What do you do? So on the first, the reason why we have shied away from taking on diseases that have genes that have, I'm going to start with highly variable penetrance, is it is likely that it is many modifiers that are making up that variation in expressivity, not one. If it was a single one, right, it could be interesting. But then you would ask, why is it so different? If it's a binary switch where it's helping, genuinely, like why is it here and then here and here? So there's the reason. Now, sitting behind that, and I might ask Yolanda again as the deep second site suppressor expert to help me here, is that we don't know what happens between that what I'd say is, very significantly variable expression, and what we've been looking for, which is highly penetrant. Because we know that for highly penetrant and that bimodal switch where it happens rarely and it flips it, that is something that is most likely to have a singular second site suppressor. When we talk to Bob Horowitz or others, they all agree on that. But what people don't know, and at least I haven't asked it well, and maybe Yolanda can help, is somewhere between variable expression in all individuals and fully penetrant. There is this place where you went, which is like 90% penetrant. What's wrong with 90% penetrant? And that came about because when we had no medical records and we couldn't go back to anyone, right? If you don't have access to medical records, we had to have something where it sort of like stood out so starkly that we didn't need to go and look at the medical records. So now that almost all these databases have a way to go and look at the symptoms, and when we went through all of us, we could go through and check, did they have a little this, a little that, and you could ask, is that like a very mild form of it? Do you see what I'm saying? That sort of opens up a little more of a window. So for now, we have set for starting it out and showing and putting the resources in the best way. Just like you said, let's do fully penetrant. But Yolanda, I think you're in a better position to comment, is there a waiting window of things that aren't fully penetrant from the work you do?
Yeah, I guess it depends on why it's not fully penetrant. You can imagine that a disease would be 90% penetrant because the suppressor is a very common variant, and so 10% of the people already carry it. And so this would be perfect. But if a disease is not so penetrant because, there's many different genes modifying it, then it can be caused by, multiple combinations of multiple alleles. And it becomes pretty complicated.
I think one thing, thank you, is that Yolanda and Maddie, I feel as if we should have a reserve pool. And Sharon, do you see what I mean? Like where people who have something, instead of saying it's an exclusion, it's like let's go into some sort of like reserve pool. Because I do think that there's enough of a reason to get excited. And Megan, and as you know, there's so many things that then it could open up during like it's there. Just one other thing, and then give it back to you. On the gain of function, because small molecule, and I'm not talking about in the second site suppressor, you have to have a loss of function that you're looking for. But for the primary gene, it doesn't matter whether it's a gain of function or a loss of function.
Oh.
Because what's happened is, let's say the gain of function is something that was an activation that caused the disease. That's what I mean by gain of function, right? So something that activated that, you could see the suppressor be something that tunes that back down. Right? So it's more that small molecules, the thing I would ask is why use a second site suppressor if it's turned up? Why not just make the drug for the gain of function in the primary? I mean, that during like this, right? So if it is a gain of function, go after using a drug to knock down the gain of function instead of going to a second site suppressor.
And if I can just do a quick follow-up, because it helps to be a little concrete. So we're looking at this genetic variant in SPINK1 in the pancreas. What my organization is focused on pancreatitis, hereditary pancreatitis, and that SPINK, there's trypsin released in the pancreas, and the SPINK1 kind of turns off the release of trypsin when there's been enough. And when that is broken, and Crystal, please correct me if I'm wrong. I have a colleague who's also the mother of a child with SPINK1 pancreatitis on with me. It's a second Megan Golden. If that's broken, then the trypsin keeps being released in the pancreas and causes all the inflammation, pain, fibrosis. So it seems like even if there's other factors involved, if you can get that, if you can get that to work to turn off the release of trypsin, it should make a difference.
Yes, just can you see my screen?
Yes.
Yes.
I can't do this for every gene, Sherry, but it just so happens, and the reason why Yolanda was smiling was SPINK1 is one of our potential candidates, and everything you just said about what it is that's there is we are interested in it because of these patients that are underrepresented. That way, so that's a clue. And our current attitude, right, beyond, I'm showing our current slide, right? So discuss with, right? But the drugability of that we think is interesting enough to pursue.
But it isn't fully penetrant.
No, but there was a reason why this was in. Can you help me with that, Yolanda?
Yeah, so I guess there's two different things, because I think, Stephen, you're mainly talking now about also finding new examples. And this
Exactly.
one was just described in the literature already. And so we just started looking at this, if people have already found something, then, we should not ignore it.
Okay.
Yeah, so did you understand that our
Yes.
sweep through healthy individuals may be hard to do when it's not, but if someone has found that already, okay, and that's what
Okay.
we want is, and I think that that's actually a really good point, Megan. I could see organizations through their own work, similar to what we just showed for SPINK1, independent of us doing the find the deep rare family or have the tools we can use to sweep through quote normal genome banks. They may have evidence from other assays that then can be matched with what happens in biobanks and tells us more that we want to know. And
Huh.
that's what we did there. So I think
Okay.
actually, Yolanda, that reminds me to talk, I should include that third arm, right? Which is things that didn't come from sweeping through genome banks, didn't come from rare families, but came from rare disease groups working up and finding something that looks as if it was interesting, which then have to be validated for how does it look in human populations, which is easy to do. And it's different than looking for de novo or sorry, new resilient individuals.
Thank you.
Other questions? Allie.
Okay, so thinking about the penetrance aspect, I think of the groups I'm aware of sort of have, a few recurrent mutations and then a whole smattering of other mutations. And so we could, to some degree, could assume like maybe those are the mutations, the recurrent ones that are maybe most important when we think about regularity and penetrance. But I feel like the penetrance question is something for most rares we don't really know
There
so
you go.
well. And
There you
maybe
go.
this is getting at that. Is that what you guys are able to figure out and looking through the greater set of genomes you have access to, that you would see where, if any of those mutations show up, and you have the EMRs to match with to understand if they're
Yeah, yeah.
actually healthy or have some kind of related, maybe not what we're seeing for the rare, but some associated phenotype that maybe would be like a sliver of a part of a syndrome
Yeah.
associated.
Yeah. So, Ali, I put down the de novo the first time question, and I've just written down on my keep notes. Um, uh, uh, is there a way to harness those on which there's less information? So, the reality, as you were pointing out, I think this applies to, P53 to CF to any disease gene. The maps of where are known mutations have virtually always more sites, more alleles, where not enough is known to know if it's fully penetrant. And so we want to specialize on those which have a high degree of information. So those are generally the more common ones, because the more common, the better the chance to know this is fully penetrant. The problem is the rare ones just don't have enough data to know whether it is. And so we have, in terms of talking of, second wave, third wave, in the first wave, we've said, let's make it highly penetrant, and that involves highly penetrant for those that are very well known. So it wipes out so many that are there, because I think that's the right thing to do. But what you were saying that will pull us off and bring in Eric and Yolanda and others is when we're doing the sweep through the 5 million genomes, is there a way to like carry along? Do you see where I'm going? Carry along, not with the primary intent, but carry along the ones where there's less knowledge of the penetrance. And we may have better maps of how well those are characterized because people haven't done just you see so there's a little bit of the beware that we don't throw out the alleles in genes that are not our drivers, but track those and how those are showing up in healthy individuals, because it could be because they end up giving information later. And I really like the idea of having a wider net with the focus where we know it's highly penetrant, but saving the other alleles for that gene and not discarding that whole thing. It's a really nice idea.
Can I ask one more question?
Sure.
So how common is probably not the right word, but how many conditions meet your criteria in terms of having a fully penetrant known variant that you can look for the second site suppressor for? Because I'm just thinking, or in our case, something that is in the literature, even if it isn't fully penetrant. I'm thinking like any patient organization would absolutely say yes to doing this, but it's more a question of finding, is it a needle in a haystack? Is it, how many of these things are there out there?
Yeah, I think we're going to have to place a constraint, not the organizations, to your point, on what is the first wave through, and get funders to agree that if we get to a proof of concept, we have a logical path to wave 2, 3. I think that's the way to do it. And I think having that constraint, I think that before, when we had 162 genes that we were taking forward, that were pretty tightly curated for fully penetrant with those 800 alleles, a lot of work went into that. And one inverse thing to do, Sharon, is to go and find those organizations, because we already have vetted those and we already know those. And how to use that versus new. But I also think that that was done 15 years ago. And I think there could be some better ones that might come in and bump some of the. Do you know what I'm saying? But I do think to answer your question, Megan, that this could go in many directions. Well, we need to get some wins in soon. Part of that is doing that priming the pump and going and showing if we get a second site suppressor candidate, we can validate it. When we have a target, we can make a small molecule to it. When we have a person who's new, we can consent to get the data. So that's sort of what this next year is about, is doing that. And I think wave 1 may be relatively tight, and what you're talking about really valuable, but might be wave 2.
Yeah, sorry, my dog decided to speak up.
Okay. We can't hear your dog, Megan. And Zoom is fabulous at screening out chainsaws
Okay,
right behind you.
So maybe I haven't seen your survey, but maybe what's needed is kind of more like an RFP, which is we are looking for organizations that have, fully penetrant variant or B,
Yeah, I think what we were thinking doing, and you're right, is we were going to do it as a 2-tiered. We were going to go through and collect all first, okay? Because if they're not interested, and then we were going to go and take those that showed up and come back to them with more information. So I think if we were to do it efficiently, I think putting in that question and not reaching out to those who didn't. But I feel as if building that community and knowing who they are and then figuring out is that wave one, wave two, and instead of only talking to the people, I think that I could see it either way, I do think that the first was, does this sound something where it's worth, I think it's going to be we need to do almost like groups of 4 or 5 at a time, organizations that we talk to on a call like this. Do you know what I mean? Like when they've said, we're interested, we'll gather 5, Megan, you know how we do that. I did that before. And so we're not going to do it one at a time, but we're not going to do 100 at a time. And let's, so thank you.
Other questions? Monique.
Can you say a little bit more about the role of the patient and the advocacy community? How much of this is sort of screening genotypes and alleles, and how much of it is actually the accurate tracking? I know you're doing work with wearables and tracking apps. How much of that, increased fidelity in the phenotype and the role that the individuals and their families, especially those with those potentially hidden, right,
Yeah.
alleles?
Yeah, such good questions. I'm going to take it to the end. Do you remember how there was that circle of steps, and the last step was doing that sort of phase 1, phase 2 trial? One of the
Yeah.
things that we've put into our proposals, and Maddie knows this well, is for non-resilient projects that for you and me does. We've been working hard for 5 years on how can you follow a state change in a disease enough at an N of 1 level that you don't need a randomized clinical trial of 200 people and aggregate data. You could work with numbers more like 30 to 50. So this is a big part of what we do, is how could you help identify a state change? And this is primarily, interestingly, not about going to the FDA. We have these people who now, as Megan knows, we've built this thing called a compass. So not the researcher, but the individual patient is doing experiments. Does this make me better? Does it make me worse? And what does? And trying to get it so that we get from a given individual, we go, if it works for you, I don't care if it doesn't work for another. So we've been building these tools that allow you to look at state changes at an N of 1 level that we intend to use when we get to where we could do a trial, because we can't do it any other way. So put that over there and then go all the way back for the organizations. And what we're looking for, we have a way to do the sweep through the biobanks with very little other than a sort of a validation of what we think has curated well is highly penetrant is. Very little. So it's not like, dozens or hundreds of hours of work. It's very minimal. Once we've found those individuals, if the organization has ways to have families that they've been following, it's very helpful. When we get to where we're trying to sort through what are the loss of function alleles, and this is where I think it's very few. Any organization that has built it so that the people they fund are ones that actually need to share their data publicly, okay? That is awesome. Okay. But as you know, Sharon, very few organizations can take their researcher at Harvard or in Texas and their organization, their university will go, forget it. I'm not making that available. It's sick. Okay. And when you want to get me like riled up, get me sick about how a group you pay for them to do the work. Okay. And then you don't have the right to share that with. And if you ask the patients, and we do this all the time, participants in our study, universally, 70% of it say share with anybody, right? That's what they say. They don't say don't let it come out of Harvard, okay? So you have the people who need it, us, you have the people who funded it, you, and you have the patients who want it, but it doesn't happen. And so the reason I'm bringing that up is that type of data where it's available is sometimes you can take it and go to the journal and say it's publicly available and I can pull it in this way. But it's almost like elders in the room. What we've learned is the rare disease groups so intimately know what's really going on and what's happening. It's the adult in the room to go, no, you shouldn't be thinking of it that way. So it's not funds. It's more shepherd us and make sure we're not doing stupid things as we're trying to do the validation. through to the end. Does that, Monique, does that?
It's very helpful. Yes, thank you.
Great, thanks. Paige.
Hi, I'm a genetic counselor at Emory, and I just am trying to think about like if I have any examples of these types of things. I'm wondering, we've talked a lot about like on the molecular level what you'd want, the penetrance level what you'd want, but I'm curious if there are any phenotype things that you'd be prioritizing. Like, do you really want neurodevelopmental disorders? Are you excluding any phenotypes? Things like that.
No, it's a great question. Two levels, and then be glad to talk to you about it later or get something into the descriptor, like we put into the survey. But basically, originally, we defined it extremely narrowly as lethal because we wanted to make sure if someone was alive, it didn't make, right? So that was how tight that 162 gene. So those were highly penetrant, and basically those particular alleles had not been seen in people growing past 20. Okay, that's how we did it because the genome banks are not taking kids at 10 and 12, right? For the most. Okay, so that was where we started. Then the ability to have a phenotype which is not lethality with medical records now. You want to have something where it's quite obvious that it's not someone that you could look at the severity of the disease because the seriousness of the normal allele is so strong that you just expect to see this in the medical record. But the last thing is that when we want to do the validation assays, it really helps to have something that we can follow at a molecular level, right? With or without that shift. And so there's some diseases where that phenotype at a in vitro level is strong enough that it's easier for us to go and do that validation. Does that make sense? But that's not prohibited. There are enough ways to do that. But did that help on the where it is?
Yeah. Yeah. So it has less to do with like the type of symptom you're seeing and more with the severity, right?
Exactly.
Yeah.
We don't
Okay.
care whether
Thank
it's
you.
CNS versus immunology versus, doesn't matter.
Okay. And Allie, last question.
Okay, so thinking about validation steps, every PAG is always wondering if they have the right resources available in animal modeling or cellular modeling. What is your group going to be looking for when it comes to that validation steps?
Yeah. The cool thing is that because the person had this allele and they had that effect, we have a different criteria for validation. So like it starts with the fact that this person was alive and they had that. So just so you know, validation is different. When you come to what's the in vitro validation models, it used to be you had to have a physiologic assay that mimicked what it was that was turning that from good to, so functional to non-functional. Now you can use various expression proteomic assays to give a portrait of what that cell is with and without the phenotype. So it's become a lot wider in terms of the in vitro assays that you can use because you also have this confidence that over here that you had some person who had it. And then the last thing to say on it is the easiest way, and I think we'll find this, that will happen occasionally is in that 5 million, if you find two individuals that have the same primary mutation and across their 200 loss of function alleles they have in common one, then it's trivial. Did you see why? If you found two examples across the 3 billion base pairs of a second site suppressor identical happening for two people who got it turned from non-functional to functional on their in terms of their first, that is almost no longer needing the type of validation that do you know what I'm saying? That's really strong evidence.
Fabulous. Well, Stephen, I think this is a great start. We got some very good questions on the table. People who are watching this webinar in the coming weeks, as we deploy the flyer and the survey, will see this, I hope, as a resource. And as you said, this is just the start. There will be a lot of conversation, and just us all working together like this will advance the cause that we're all so passionate about. So thank you so much for your continued dedication to this over many, many decades. And I look forward to us, and the us is big, working together.
We do too. And thank you all. Hope to connect in the future.
Wonderful. Thank you.
Thank
Have
you.
a good day, everyone.
Right, right.
Bye-bye.
Thank you, Sharon.
Bye-bye.